Once upon a time, in a place not too far from here, in a time not too long ago, there was a most intelligent professor, Dr. Fei Wen, who had a brilliant idea of a protein that could change the world. You see, labs across the whole globe have been coming up with new and novel artificial antigen presenting systems. That is, systems that stimulate a type of white blood cell called T cells, just like infected body cells do. Dr. Wen's idea was for an artificial antigen presenting system that would be different. It was new. It could be something amazing. But Dr. Wen got really sad because she realized that her artificial antigen presenting system proteins didn't even exist. Dr. Wen gathered together a group of students who were educated, yet teachable, full of enthusiasm and eager to conquer the task of creating these proteins. I, Stephanie Tolbert, have the privilege of participating in this wondrous endeavor. This artificial antigen presenting system would require a series of proteins working together. The details of the system remain top secret until the long-awaited publication is finally finished. However, I have been granted permission to reveal the process through which we create our top secret proteins. For ease of your understanding, I have created this nifty flow chart of our process. In order to create our protein, we first need a plasmid containing DNA for our target sequence. And in order to get this cDNA fragment, we need to isolate mRNA from the cell. But before we do any of this, we must first design a precise and accurate plan for our final protein construct. To create the plan, I spent hours and days and weeks on databases understanding proteins other labs have designed and searching for the DNA sequences that will work just right for our system. Once we have an excellent plan, we can enter into the lab and begin our work. First, we take cell lines expressing the proteins from which the elements of our protein are derived, and we extract their mRNA, getting a small tube containing total mRNA, which is all of the mRNA from the given cells. Using reverse transcriptase, we create total cDNA, and through polymerase chain reactions, PCR, we amplify only the desired fragments of DNA. Then we isolate those fragments by using electrophoresis gels and gel extraction kits. Next, we digest those amplified extracted DNA fragments with restriction enzymes to create sticky ends on the DNA. We also digest a given engineered plasmid with those same restriction enzymes so the sticky ends of the DNA fragment and the plasmid match. Our digested fragments require even more gels and extractions. Using an enzyme called T4 ligase, we ligate them together to form our engineered plasmid, containing the DNA insert encoding the sequence for our protein. This ligation mixture we then transform into Mach1 cells. Due to the properties of the plasmid and the properties of Mach1 itself, each individual colony will contain copies of one single plasmid, and most colonies that grow should contain the target DNA sequence. We pick colonies off the petri dishes from the transformed ligation mixture. After growing them up overnight, we use a mini prep kit to extract the plasmids. We transform the plasmid one more time into Mach1, so Mach1 will amplify our plasmid, and as well as safely store a stock of our plasmid in case we run out. With another mini prep kit, we extract our plasmid from the Mach1 cells so we can sequence that plasmid and also transform the target DNA into other cells, which will express the desired protein. But before I move into the protein expression, I'll take a couple steps back and explain to you a second method of creating plasmids called homologous recombination. With this method, we similarly take the digested plasmid but use only undigested PCR-amplified DNA fragments which get co-transformed into yeast cells. The digested plasmid and DNA fragments are designed to have overlapping ends which anneal and form our plasmid within the yeast cell. We similarly extract the plasmid from the yeast cells and transform the plasmid into Mach1 in order to amplify and safely store our precious plasmid, which contains the target DNA required for our top secret protein. And so we finally return to the point of having this plasmid sequenced and ready. There's copious options of what we could do with our dear friend, Mr. Plasmid, but I will explore with you 3 different routes I've personally traveled in lab. First is the good old simple bacteria to express our long-anticipated protein, but bacteria is only good for expressing simplest proteins. Once a plasmid containing our target sequence is transformed into bacteria strain BL21, the bacteria cells are induced and will express the desired protein. For creating more complex proteins, more complex cell machinery is required, so we can step up to the yeast cells. Similarly, the plasmid is transformed into yeast strain L2612 and the cells express the desired protein. For even more complex protein constructs, insect cell baculovirus expression systems can be used. First is a transfection of our plasmid and linearized baculovirus DNA into an SF9 cell. This cell will create a virus containing our target DNA. This virus can be amplified by infecting new SF9 cell culture. The amplified virus is used to infect high-5 cells which express the desired protein. So, those are a few of the different methods for engineering proteins. I hope you enjoyed the video. Thank you to Sher for the funding. Thank you, Dr. Wen, for another great summer. Thank you to my coworkers, the Wen Labites, for your partnership.